to and during the pandemic move across the country to a new job and arrive at a campus that is empty of any people and devoid of any social activities. So Raziel has gotten to know members of my lab group pretty well, but we wanted to take this opportunity for him to introduce himself and introduce his work to the rest of the department so that hopefully next spring and summer when the pandemic clears up and we can all start to see each other again, you all know who this new guy is in the department. So in addition to just introducing him as a new member of our department, we thought some of the background research or previous research that he's done in previous positions would be of real interest to members of the department. So he's got a seminar today on, he's going to cover some of his work in his previous postdoc position at Iowa State on root growth in the Midwest. He's going to tell us a little bit about some of what his projects will be here at Penn State with my lab group. And it's kind of an opportunity for us to introduce the projects he's working on and see if there's potential for collaborations with other faculty members and lab groups in the department. So Raziel is going to tell us a little bit more about his background, but immediately prior to coming to Penn State, he did a postdoc for 4 years at Iowa State, and we'll hear a lot about the work he did there. He did his PhD in crop ecophysiology at the University of Lleida in Spain. He also did his master's degree at University of Lleida, and his bachelor's degree is from the Agricultural University Antonio Naro in Mexico. And he also worked for a year as a crop breeder at CIMMYT in Mexico. So he's got a really nice background, diverse background in different plant science topics. And it's my pleasure to turn it over to you, Raziel, for the seminar. So go ahead. Thanks.
Okay. Thanks, Charlie, for your introduction. It's pretty good. Let me see. Now it's full screen.
We're seeing your presenter view now, Raziel.
Presenter view now. Let me change this. So just going here. Seems has some problem to connect it here. You see now?
So we're seeing your program now, not necessarily the slideshow, just the PowerPoint program.
Since it's going wrong here.
The joys of having 2 monitors on Zoom.
Now can you see it?
Yeah, there we go. That looks good to me now. Yep.
So it's the full screen, right?
Yes. Yep.
Okay, perfect. Sorry for the inconvenience. Always to have a couple of monitors create double problems. Okay, so thank you, Charlie, for your introduction and also for the opportunity to present this seminar. And thanks to Robert for extending the invitation. So a couple of months ago, I arrived here at Penn State. And today, it's my pleasure to deliver this talk, as Charlie mentioned before. And for me, I took this opportunity to introduce myself because the current pandemic doesn't allow us to be like face-to-face to share our experience. So at least I had this opportunity, like being new here in Penn State. And I took that like great opportunity to be able to introduce myself since that I'm doing. So my presentation, the way I frame my presentation is to give you a little bit of background of my research and to talk about some challenges that we're facing for the crop productions, root distribution. And also I found that it was important also to talk a bit about the interaction of nitrogen with the root systems. And at the end of my presentation, I will also to talk about things that I had been doing here for the last 2 months. So who am I? I am a crop physiologist. I want to mention that crop physiologist working with these four beautiful crops: corn, beans, and rice, wheat. So I had some experience on that. What do I do? So what I do is to improve crop productivity and crop system performance. All my work has been around these topics, and I want to share with you also that, as Charlie mentioned before, my background is in plant breeding and genetics. This was my degree at the University Antonio Rara, Mexico. So having this background in plant breeding, I was working with this little boy here, this corn, and very short, like short plants. That was my job that I did there. So develop new inbred lines that will be able to cope for the climate change in the future. So this, having this background and opened me the opportunity to join CIMIT Mexico. I had the opportunity to work with Professor Jose Luis Arau. I was a breeder during this time, working in wheat and maize, selecting varieties that will be tolerant to drought stress and low nitrogen, um, and soil with low nitrogen. So in this picture, we can see that Jose Luis Arauz, and also in the bottom, we can see Professor Richard from Australia, one of our many meetings that we had during this time. And then I moved to Spain, where I did my master. I was working with Professor Gustavo Slaffer. So my work in Spain was related to crop physiology, particularly in maize. And we were growing maize in different locations, evaluating different varieties, different hybrids. And the goal there was to identify those varieties that had some characteristic that can cope with heat stress. That because was the idea that in the future the temperature will increase. So now we need to be able to find new places or explore new opportunities where we can grow more crops in the eventual if we had to face these situations. So my work was very successful, and I got some good answers to many questions that came up in that time. So this led me to go for a PhD, and I continue working with the same professor. So I think he was happy, I was happy with him, and we work in very innovative project that was also testing different temperatures direct in the field. In this picture, you can see clearly how we're building the chambers in the field and testing different levels of nitrogen. That because the theory said, or many work from people around the world, is telling us that, and also based on the projections, the temperature will increase in the future and for the in the hundreds. So my idea, and also Gustavo's idea at the time, is like to explore that mechanism that the plant will, or these changes that plant will have as the response in adaptation and mitigation to these weather changes in the future. So that, I also want to mention a little bit that I also contribute to work, very interesting and very good job. I also had the opportunity to work in projects that I think is very well recognized, because I was, since I was in CIMIT Mexico, I was developing a drought tolerant maize that can cope with many issues and many problems, but especially for droughts in some countries in Africa and also in Latin America. And then I also had the opportunity to work with international groups that were working and developing new varieties of rice that can express also proteins against HIV. So this was the most recent things that we had done. I worked with many people in Spain and the UK, and of course here in the US. So it was very international collaboration to be able to develop this work. So this is kind of my background since I've been doing, so how I'm interacting with different people and also how I interact with different crops. So the goal here is to be able to improve crop productivity in the future and also make sure and to understand how these changes will be in the plants. So with that ideas in mind, also, I want to mention that I don't see plants as everything because always has a connection with different factors. So in the previous work that I did during my master and my PhD was just related above ground. So everybody was asking me, do you know about plant? I was saying yes. But then I was thinking very serious to say, no, I don't know the plants very well because I still don't know what's going below ground. That is the second part of the plant or the half part of the plants. So which I would say that it plays very interesting role also in the plant productivity because we focus mostly in the above ground, but the below ground, we don't know what's going on. So when I joined Iowa State was specifically to work in that area, which is the above and below ground. And the goal to that was to be able to improve crop modeling, which is absent that is based on Australia, but also Iowa State is part of this initiative right now. So I took over this area, which was the crop above and below ground, and to be able to develop functions that will be able to improve and be able to forecast crops across the state, but at the experimental level. So as the time passed, we were able to, or not right now, we are able to predict things at the cropping system levels across the Midwest. So different states in the Midwest. So with that, I was introduced to, when I arrived to Iowa, I was introduced to a very diverse place in crop productivity. So because we had corn and soybeans. So corn and soybeans is planted from Illinois to Nebraska, from Minnesota to Kansas. So we can see the diversity of crop that we had there, only two crops, of course. So Iowa is one of the most important producer in the United States. So I was very happy working with a very diverse crops. And so before that, I want to go into more detail and what is the challenges in the productions system around the world. So one of the things that I found that was very important when I arrived to Iowa State was like I was always working in stress like drought and low nitrogen, but I found out that it was also interesting that in the Midwest there exists one of I would say a problem because actually it's a problem that is the shallow groundwater. And the shallow groundwater is not just a problem in the US, but it's also a problem at the global scales. So we can see in these maps in the blue color how the groundwater table changes or how these behave across the globe. So in Iowa, after that, we did reading a lot of the literature, and we found that the water table was one of the biggest issues there that because constrained roots to grow. And we developed a study that we were measuring the root depth and to see if there was correlation between the depth to water table and the maximum root depth. And yes, the answer was yes, there is a problem because the water table was pretty shallow. You can see in this graph that the water table range about 70 centimeter, and also the deeper will be more than 2 meters, but still is shallow. So you can imagine the problem that farmers are facing there when the water tables are changes across the year. So here is a scenarios how the water table works in Iowa. So in the early in the spring, we can see that the crops are suffering because the excess of rains or the excess of water at the very beginning. And farmers has to develop new strategies how to grow plants in their ground. And so they had to install tall drain to be able to remove this water that occasionally will bring some problems to the crop. So it's very typical that the farmer, after when they harvest their crop, will come over to the field and start installing tile drain to be able to remove this water that for the following season will reduce the crop yields. And also this problem can bring more and more problems because this also contribute to the nitrogen leachings, and also this change the ecosystem, all the nutrients that remain in the fields after harvesting, so it can be removed from the water tables too. And this changes a lot of practice for the future crop in the following year. So with this idea behind, we were thinking what we can do and what we can improve to that. So I started looking in the literature and then I found no much information. And whatever information was about root systems in Iowa was in the early 80s. That nowadays, since it had changed a lot, crops are changing, varieties are changing. Some of them are already developed some tolerance, but there was no study that is showing what things have been done. So with that, we focus on to study the roots. And this presentation, I will be basically talking in these two papers. That one will be the distribution across our profiling. The other will be related to the nitrogen, the relationship between nitrogen and root growth. So for that, many questions came up to the table. So one of them, or the most important for me, was why the studies and roots are important. So one of the things is because the roots can allow us to understand better about the relationship between plant and weather, and then also to be able to predict yields and to understand the nitrogen and carbon cycle in the ecosystem, and to develop, and of course, to develop algorithms for modeling, either roots or cropping system model, biophysical model, which is important. So with that scenarios, we start thinking about the root, how the root will grow when we have this type of environment. One was the question, what the root, how the root would perform when you had shallow water table, and when you had a deep water table. The deep water table is no more than 2.5 meters, so it's still very shallow. But this fluctuation over the season creates some problem to the crop, and basically this problem is related to the root system. So on this map, I'm showing that our fields that were distributed across Iowa, and how the water table fluctuates also. It represents basically how the water table perform across the Midwest. So I'm not kidding, that is the actual picture of the water table in the Midwest. And probably also here in the East Coast, in New York area, they also have some, I know that there is also some problem with the water table, and also they are installing in the past, they were installing in the past some tile drains. Obviously, we need to monitor it. We were monitoring the water table across the season, the all year long. We were installing some wells and to be able to follow this trend of the water table across of the season. So here, I'm presenting to you how my approach was used to be able to measure root depth. So we were using one simple probe. So we were pushing in the soil, and we were going like 2 meters deep using this. So if I want to do this for sure here in Pennsylvania, it's not possible to do it for sure. But in Iowa, because the soils are deep and it's possible to be able to go 2 meters easily. So this we were using also the hydraulic probe to be able to collect the deep soil core that we were using to as our root systems. Here, this is our the cleaning process. We were using the Gleason system that consists in putting water with pressure and air in pressure, and to remove all the organic matter and all the soils, and to get the root system, all the pieces of roots in clean versions. And here you can see how we had all the roots. We had thousands, I think for now it's more than 500 samples that I processed with a wonderful team that I had working with there in Iowa State. So we had plenty of samples. We were using the WinRhizome. During all this time, I was using basically WinRhizome. There was no other source for me because I had the opportunity to work also with people in the USDA, and they had a facility for the roots. And here you can see when I'm analyzing, scanning my roots. And of course, at the end of everything, roots is important not only to evaluate how plant perform, how plant growth, but also it's important to know how much of these roots will contribute to the soil system. So we were measuring also carbon and nitrogen and all the tissues. In all the samples, we were evaluating or we were testing how much nitrogen, how much carbon we had, because as the soil science point of view, and this is one very important information that we always have to have, especially when we are looking at the things to the system approach. So, and then this will be sharing, for the next slide, I will be sharing how the root system will behave in the soils. And particularly, I just focus on two of the components. Okay, I consider that they are the most important, which is the root length and the root mass. So everything around that is just calculation from that. So I don't want to go very specific in many of them, or many of the traits that we can have from the root, but I want just focus on root mass for this presentation. So I just want to tell you that I measured, I think as far as I remember, it was more than 50 traits, but for this presentation, I choose to talk only about root mass and root length. So here in this picture, we can see how the root mass this is distributed in the growth across soil profile. You can see in the left panel how actually this is the information that we got across all the sites that we were working. So in the right side, you can see that we have the blue color correspond to the in-row sampling. In the picture, you can see what is the in-row that was exactly in the row of the plant row. And all the yellow color is in the middle of between two rows. So we measure the root mass across of these 2 points, and then we get the distribution of this across profile. So around 80% of the root is distributed in the 60 centimeter. But interesting is that the root also can get about 150 centimeter. So this root, the deep roots, and I believe that in Iowa is the ones that are doing a good job to trying to capture all this nitrogen, all the nutrient that is leaching through the water table. So I believe that deep root, they can do a good job getting this up and to reduce a little bit the nitrogen that will be lost through the water runoff. So the same thing happened with the root length. So the distribution is more or less the same. The amount is the same. 80% is in the top 60 centimeter. So one of the important things here, what I tried to show was that is up to every research what is the objective that you want to answer. So you will choose where you want to go or how deep you want to go. So because especially when we talk about root mass, which is the main driver, of the soil carbons, or the major contribution of soil carbon, which is also related to the microbial activity and all the things. So it's like big community inside of the soil system that at the end of the day that we will be able to understand how much of the roots will be in the soil and how much that will be worked. So that was important. So we would see the variation in both components here, and this variation is across because we had different type of soils, different type of management. So we can see clearly here how that changed. So this obviously will also give that, will give me differences in the plant growth and plant performance. So what it could be expressing basically in the root to shoot. So in this graph, what I'm showing here is that across all the experiments that we had, I determined the root to shoot ratio, which is actually the ratio between the above and below ground dry weight. So the range that I got is in green color. So the means was about 10% of the plant. And the blue color represent, I also looked in the literature, wherever it was already published, the literature. I did like very deep searching of data. So, and then I did some calculation. I calculated the means, median, and third quartile, and 90% confidence. So why I did this? Because the reason, the explanation to that was like many work, many research, especially looking at the soil, the root carbon contribution to soil, basically they use a single value. So with this distribution of my data, what it's telling me here is like, okay, you can choose one value, but you tell me which one is the most precise or the more accurate that will be able to estimate better the amount of root that you will put or you will estimate. Because when we calculate the root mass, also we can probably be overestimating or underestimating our root carbons. So in the panel in the right, I use all these parameters with my observed biomass, and I did the calculations, and the above biomass using these parameters to be able to estimate root biomass. So it's kind of like to say, if you look in the literature, get the root to shoot value that you find over there, and use your biomass that usually is the only thing that we measure in the field, and then tell me how to predict or which one you will predict. So in this paper, I'm not telling you, I'm not offering you a number that you will be used, but I just want you to be aware that using a single value from the literature can yield in different values of root mass. So in the green color are the values that I collect, my measured biomass and my measured root mass too. So the variation is shorter than what we had in the literature. In the literature, we get very, very large range of variations. While in my data, using first of all, like consistent protocol for sampling root and for sampling above ground, so we can see the variation is shorter and the literature is large. But also for that, it's also important to know what are the driver effects of the root system. So we found that obviously it's gonna be the water table for corn, and also all the parameters or the variables related to weather. So this is, in corn at least, what caused the biggest problem: weather conditions, weather variable. In soybeans, it's a bit different. Obviously, the reason of that is because corn and soybean had different root system. So in maize had different class of roots, while in soybean had a taproot. And sometimes I believe that the relationship with the water table was low because as we know, the soybeans or beans or legumes had opportunity to develop parenchyma, which basically is one of the source of the changes that the plants suffer in order to be able to survive under stress conditions. So I don't want to go very much into detail, but for sure you can see in our papers and with more details about all the processes that happen there. But just I want to let you know some, or to share with you this information. So of course, I think you are wondering now that how this guy calculated the root mass, because as I showed you in the previous slide, I was getting root data from in-row and between row. So I don't want to miss the opportunity to introduce also and to let you know that I developed an algorithm that I can account for the sampling point. So this algorithm that I create is just the weighted average, which use the measured root value, and also it needs as an input the sampling positions. So to develop these equations, we create sampling points in between the row and the distance between the plant and the middle of the row. So we were sampling different points and to create an equation that will account for that variation across the between row and in row. So, and then all the data that we were collecting, we use this equation to be able to get one single value that will represent the root mass, because it's difficult to think. Also, when you see in the literature that there are different values that has been provided, but sometimes it's very difficult to know which one will be the best or the most accurate to be able to calculate root parameters. So with that, I will pass to the next section that is going to be related to the nitrogen. How the nitrogen interact with the root growth. This is an experiment that we developed also in Iowa State. This picture actually is one of my experiments that I did while we ran it. Here you can see clearly how the difference are between fertilized and unfertilized treatment. So I had the opportunity, I was pretty lucky, I would say, because the group that we're working with, we ran experiment just specifically to look for root systems. So we were running different experiments and the conditions were exactly like that. So I also had one of my, I will call my hobbies, like to take pictures. I like to picture to get a shot of my experiment, my plants, everything. And actually this is one of the pictures that we got when the plant was growing. So to be able to detect difference between crops, between treatments, sorry. So for that, there were 4 sites, and I used 2 different levels of nitrogen. And also my experiment included different types of soils. So this experiment, for sure, I was not expecting something different when we look to the yields. So as everything related to nitrogen, especially in the grain crops, particularly a corn, that you put more nitrogen, you get more yields. But this yield will be until a specific value that will reach the plateau, and then it tends to decline. So in my experiment, I found also this response, more nitrogen, more yields. But one interesting thing, like, okay, we always think that everything will be perfect, but actually the nature saw that no. Sometimes we expect things, but it happened completely different with what we were expecting. So it was the case of that side that there was no nitrogen response in terms of yield. So everything looks the same. So no matter the amount of nitrogen I put in the soil, but the response will be the same, which was magnate that had high clay content. So that high clay content is, I would say, and in terms of soil, is one of the soils that had high response when you had drought stress, when you had, dry condition, when you had wet conditions. So there are more fluctuation in these type of soils. The other soils was loamings in 2 of my sites. So these sites were actually were the more productive areas in Iowa. The Muscatine was a site that is very close to the Mississippi River, that obviously is sandy soils. So it's quite different. So we wanted to do this experiment to be able to evaluate what the effect of nitrogen will be under all these type of soil in all the growing conditions. So one interesting thing here that we found was like opposite what happened with the above ground, the below ground is completely different. So there are increments of the root biomass when we apply more nitrogen. And then when we had high nitrogen, the values decline. So that I think was one of the very interesting findings that we had in this experiment, because what it's telling me here is that the root growth had an optimal nitrogen rate. So when it peaked the optimal nitrogen rate, also the root biomass maximized to that level. So, and then after of that, there is no more growth. And actually, and which is very logical to my understanding, because the plants, they don't need to produce much root when the conditions are favorable to be able to get all the nutrients from soil and put it up in the plants that will be able to produce more yields. So I think this explanation for that was more or less related to that. And also, and here, obviously, in the low nitrogen, we found low root mass, which is pretty much expecting that. So we don't expect that will be much of growth when plants are on the low and under stress. So the root to shoot ratio is actually indicating that the response when we have more nitrogen, so the root to shoot also decline. So it's telling me that the plants are getting more yields. All the carbon are put in the economically important part, that is the grain. So, or to build biomass, at the end of the day would be also more yields, because as we know, the biomass is very much correlated with the final yields. So for that, also we did some studies, actually with my data, we calculate the root to shoot, as we know, is root biomass and final yields are not correlated. And also basis on that, so it's difficult to predict final yields basis on the root to shoot or basis on the root biomass. So for that, we create an equations that account for this variation. So are the boundary bases in our data that we collect. And this is in the cross is also on the study that we developed. So all of them is measured data. Is using our protocol, and we can see that there is a correlation. Some of the site they had correlation, but some other not. So this is basically telling me that we need to be careful when we choose a single value to predict things, because this is what we find, or what we actually happen, that there is always variation. There is no a single value that we can use to be able to predict things. So that allowed me to create this boundary and say, at least to be able to tell researchers, hey, you need to be able to predict well, so you might be able to use the boundaries and see what the distribution of the points is gonna be. So that is part of my thing that I did related to nitrogen and also related to the root distribution. But I feel that there is also an important thing that we need to think about, how long this root is gonna take to decompose. Because as we know, plants allocate carbon to the system, to the soil, but we don't know how long that it will take to decompose. So for that, I developed also one very interesting, very cool study. So in these fields, actually there are many fields that I did this study, but I'm presenting only one. And in the 2017 growing season, early in the season, what we did was to mark this area. There are many replications. For the experiment, we mark this area because it's a place that we were growing year over year over year. So we mark this area, and also we put the GPS points where the next sampling will be. So we keep this area without plants 4 years long until the 2020. So it's a bit sad that I was not able to collect the final data for this because 2020 was the pandemic situation. So I don't know actually how much of the root stay in the soil. But what I know that at least in the 700 days that this experiment was in the ground, So we can see how the root decompose. So I just want to mention, to be clear, that we didn't have any plants growing there since 2017. It was like clean area. I was taking off all the weed that was growing there, everything, like not to have contamination from other plants. And I was collecting this soil core there and measuring the root biomass in how long it takes to decompose. So I did this for all the layers that I mentioned before, but for this presentation, I want just to show you the more um, opposite place depths. It was the 0 to 30 and the 6 to 90. So you can see here how the root decline over time. So in the above, and the layer in the top, it takes less than a year to decompose, about 280 days to decompose 50% of the mass, while in the deep profile, it will take a bit longer than a year to decompose 50%. But the 90% after was my last value that I got from this experiment in the fall 2019. As as I said, I couldn't measure in the 2020. So this is how the root decompose. Maybe this variation in the composition and the timing between the difference between layers is because the difference in the microbial community that can be related to the decomposition process. So these information are very useful, all the information that I just shared with you. And that data was used to calibrate different cropping system model, but in this case particularly was for APSIN. APSIN is a model that we use in Iowa State. So that model is implemented for different sites, for different conditions across the Midwest. So we can see here that how well simulate root growth. So that is one of the important. In the left side, you can see many variables that account in order to be able to predict well. I just keep that in mind that sometimes to be able to predict things, you need more and a lot of variables. It's not just need one single value from any variable. So you just need to account for many more. So that yield is also in developing different decision-making tools. And this is the case of the prediction of yields across the season. So we start simulating the plant growth or the yield from early in May until the end. So you can see how good or how pretty okay we're simulating all the yields. So with that, I think it's something that I was doing in Iowa State, and I believe that to share with you all this information, maybe they can open more questions, and maybe we can be able to collaborate here at Penn State and open new projects to continue doing more and more to these topics. So just the take message for this part will be that the knowledge of root depth is critical to understand complex soils and plant processes, and also is telling us that the water table is the real problem in this area. And also we can see that the nitrogen, the root mass increase when we put more nitrogen to the soil, and it maximized to the intermediate nitrogen rate. And also the relationship between these variables was important for the future or open future research question or research efforts. So it's also important for me to acknowledge that in Iowa State, I had the opportunity to work with wonderful teams. I was working into lab, Mike Castellano and also Sotiris Argentulis, that I had good collaboration with all of them. And we did all this work very good. And I believe that we are continuing collaborating and doing more and more things to that topic. So this is closing my work at Iowa State. So I'll bring it to the Penn State. So I arrived to State College early in October to State College. So I came here because it's I was offered to work in the on-farm nitrogen experiments looking at corn. So obviously I was looking, I had to look for information, and this is how the yields or how the evolution of crops, in the case of corn in the Penn State, across the state, sorry. And this was one of the experiments that we had to run, but I chose to work in soybeans for at least for this presentation, show you how we were doing some things here, some cool stuff. And this looking at root system. And I measure root depths in all the things. And then I questioned myself, so how the root could be able to predict zones, productivity zones. So I look for all the information that is very important, because I also had in mind that to have the cropping system, the system level approach is also very interesting in order to be able to predict things. So I looked for the soil condition, for the soil types, and also for the land, how it's occupied for different crops. And here we can see the temperature, how it was this year. So it's telling me that in the east side of the state was more, sorry, this is the precipitation in the east of the state was more low, and in the west was a bit more wet. So this is the variation of temperature, the precipitation that we had over the state. So this was how we characterize our fields, the soybean fields. So this was imaginary images that were taken over 3 years, satellite images, and then with some work utilizing GIS systems or resources, they came up that some specific zones. So where this little square here, we can see how these include different zone productivity zones. So I sample there, I collect all the yields from this area. I also measure a lot of yield components related to soybeans, and I characterize the yields that we got from there. So here is actually the picture that is telling me how this soil looks like. And also we see the quality of the grain that we get. So I believe that any crop that you plant in this area, your seed quality will be low. So we need to work hard in order to be able to increase the quality of grain. So for that, we need to do better management. And also we need to be able to create agronomic programs that will be able to count for these things. So here in the next slide, we're showing to you how, in my mind, the root system grow in that area. So obviously I measure these values, and actually that is what is happening. So in the low part of the fields, the root system is deeper and plants they have very well plants. And in the top of the hill, they look more small with shallow root depth, shallow root systems. So I collect this information, I measure in the field this data, and here we can see the correlation that I got from this information. So it's pretty good, pretty strong. So I got this equation. I use also the planting area yield data and root depth from the publicly available data from Surgos. So, and then I did some processes using GIS tools to be able to transform this equation and use this data, and also at the end of the day, to be able to detect productivity zones in Pennsylvania. So this study that I did led me to, or allowed me to calculate the productive zones. This is the case of soybean. Don't forget that it was only soybean work. So with that equation, with the root depth, allow me to detect a productive zones across the state. So in this figure, you can see from the left to the right. So in the right, it's telling me that these are the counties. This is the list of counties across the state, all 67 counties. So this here from the white to red is exactly the site that I was working collecting corn data. So you can see that here the low productivity zones are they have very low productivity zones. What it's telling me here that they are very good soils. They are high yields. while the red here are the poor soils. The farmers are growing soybeans there, but it's telling me that about 70% of these soils are poor. So what this means that the yields are pretty low. So with that in mind, for me, it was like I started questioning myself, and then I said, can we do that also for Pennsylvania? Or we can do also for the whole regions? And my answer for that is yes, because I did this work in Iowa State, and I know that this is also possible to do in this area of the US. So the only thing I hear is like it requires more high quality data with better resolutions, and also we will be able to do things in that way. So this is kind of my approach that I propose that it could be possible to do it, continue to looking for the three major components, genetics, environment, and management. Also, we can continue to measure different factors, including management, also weather. So to look above and below ground, that is the correlation or the relationship with the soils. In the above ground, we can see the yield components, nitrogen cycle in the soil part, and the below ground, we can see root components. This using different models that also I'm very familiar with this part. We can continue using applied science methods or technology, can use the models, machine learning, and at the end of the day, we will be able to improve our productivity, or we will be able to improve different stuff that farmers are doing. So this apply for not only for corn, not only for beans, but also for small grain crops. So with that, I also want to acknowledge all the collaborators that I had, farmers, grad students, and also some extensionists that are involved in this project, and our founders, and all the agencies that contribute to this work. With that, I will take any questions if you have it.
All right, thanks very much, Raziel. That was a great seminar. So I think we can probably take a minute or two of questions for folks who are still around. Feel free to type those into the chat box. I'll monitor that. Or if you have a question you want to say on your microphone, feel free that way too. And Raziel, maybe do you want to stop screen sharing? We can see everyone's faces. Great.
Let's see, we are there.
Heather, I see you unmuted. You have a question?
Well, yeah, if no one else is going to ask one. That was really interesting. Thank you. Really fascinating, your root to shoot ratio differences. And I just wondered, when you first put up the first figure, of course, the water was identified as really important, but were those also high fertility soils that had good soil textures for plant growth? Could that also contribute to why your root to shoot ratio measurements were much lower than what's in the literature?
So one of the things that we discussed about that was that the literature, because there are not many work recently nowadays related to that the traits. So one of the reasons was that in that literature data, there are also including all information from the past, and also include different sampling methods. Well, in the literature, what we look only for those studies that were sampling at the same time, or at least around the sampling that we did, to be able to be fairly comparable, at least in the sampling time. So the difference, it can be because of, as I saw you in the question, that you can get a lot of variation in root to shoot based on the sampling positions. So that we believe that also related to that part.
Okay, thank you. And maybe just following up on that, when you looked at what predicted with that Pearson correlation figure, were those separate correlations? That wasn't like a random forest where you evaluated what were the best suite of predictors?
In which Are you talking about the maize section?
With the water table being most critical for maize.
Oh, yeah. So that was a simple correlation analysis that I presented you. But we also did, we use machine learning techniques to be able to see which one will be able to select as the most important factor. So I just show here in this figure because I was believing that was just most important to show you how the correlations are between them, but also we use the Lasso's equation to be able to detect what is the order of importance of each parameter. So yeah, it's also related to the machine learning techniques.
Good. Thank you for that question, Heather. Any other questions from the audience? I'll pose one for you, Raziel. So I was really interested and impressed by what you were able to do in a short amount of time with data at your fingertips in terms of your analysis for Pennsylvania. And I was curious about the Surgo data and the rooting depth. You showed a map that kind of categorized it, I think 0 to 80 and 80 to something or other. But what, I guess I was curious, what resolution is that data available actually at? Is it resolution at the soil type level? And then as you looked at that data, did they give it to you in centimeters down to, every 5 centimeters, or is it kind of ranges from every 20 centimeters? What are the increments? What do you think is the accuracy of that data in terms of being able to predict crop yields?
So that's a really good question, Charlie. Yeah, as we know in the Surgo data that they provide and information that is based in different study, different way of measuring the land across the landscape. So this in Surgo you can find information as the county levels or regional scales on everything. So the resolution of that data, it was at the county level. That what I use is the county level, but it's the average of across every single points that they had measured from where this information was estimated. So the resolution, I believe that at least is the only source of data that we can have access right now. And I believe that the resolution is good enough to do things. And Surgo data is being used in different modeling work, and the accuracy is pretty good. I cannot say exactly what is the resolution per se, but the only thing I can tell you that it works well.
Good, thank you. So does anyone else have a question for Raziel? All right, well, thank you again, Raziel. Let's give Raziel a digital round of applause for a great seminar. We are really glad that you've joined the department, and it's been great to work with you over the last couple of months. We're looking forward to a great couple more years together here, advancing production systems in Pennsylvania. So I appreciate that and hope everyone has a great day, have safe holiday season, and we'll talk to you all later. All right, thanks everyone.
Okay, thank you, Charlie.